Hi everyone, welcome to the last lecture of cell and molecular biology. I can't believe we're at the end of the semester already. So the last topic I have today, super cheery, stem cells and cancer. So before I get into the content, I'm going to go over some announcements, just some general housekeeping, and then I'll get on to the last lecture. So some announcements. We'll go over stem cells. I'll answer your questions you guys posed in lab, so thank you for submitting those. They were really great. And then we'll talk about what cancer is, how it's formed, and how we treat it. Okay, so I know you guys have been anxiously awaiting the final points and things to be graded. So instead of the 1,000 we were originally going to end with, I have the course being out of 850 points. So part of that was we lost opportunities for discussions. That would have been another opportunity, and just exams and things. So the final points are out of 850. The information about the final has already been posted. So instead of an exam, it's a reflection assignment. So there's 4 sections to that. All of those are in that document on Moodle, worth 60 points total. So I think everyone should do really well on that. I'm really interested in seeing what your thoughts are about this semester. And then just to make it work out evenly, I have 25 points out of those 850, and everyone will get 25 out of 25. So just for persevering and doing so well and being so on top of everything, you guys get those 25 free points. So I'm finishing up grading. I'll have your metabolism essays, if they're not done already, done very soon, and your feedback for your outlines done by Friday night, depending on when you're listening to this. So you'll have everything you need from me by the end of today, Friday, and you'll be all set for the rest of the semester. So the other thing I wanted to remind you guys of is that staff reflection. So that controversy surrounding stem cells in a lab in Japan, that reflection assignment is due this Sunday. And again, if you have extenuating circumstances, need a little bit more time, totally fine. But if you're looking for it on Moodle, it's under week 13. So where you would submit that is under week 13, even though it's due this coming Sunday. So that satisfies one of the blueprint requirements. I'll have the portal or the way you should submit your presentation up very soon, due Wednesday, the day we would have had our final. So just one more presentation. It sounds like people have had a lot at the end of the semester, and one reflection assignment, your final activity, and you're done with cell and molecular biology. Okay, any questions about that, let me know. We're going to get into stem cells and cancer. So one more time, thank you guys for doing that lab. It sounds like that was something that a lot of people got out of. You guys got a lot out of that activity, and your questions were really excellent. So I think I've addressed them all here, but if I didn't get to yours, let me know and I'll answer it for you. So some of them were very specific to the lab activity itself. So I'm going to go over those first, and then I'm going to take a step back and kind of go over what stem cells are as a whole and why they're useful, why they're helpful. So one question I had was, how are the cells themselves isolated and removed? So a lot of the steps you did when you were clicking through the lab was just washing the plates and getting it ready. So I found a video that goes over how you would do that if you were in the lab. So if you're curious, you can watch that video. But the TLDR version is you prepare those wells with that matrix, so that the stem cells are stuck to that plate. And so when it's time to take some of those cells and put them in a new plate, you have some heated buffer and other things that you pipette onto them. You kind of swirl it around, and then you are able to pipette those cells and put them in a new place. Again, if you want more specifics, you can check out that video, but essentially it's just the pipetting back and forth. One question I had was, why did the colonies change color from day 1 to day 5? So I think this is in reference to the step kind of in the middle where you started a colony and you watched it progress. It seemed like it went from kind of a black background to more yellow or blue. I don't know why the color in the background changed so much. It wasn't like a stain or anything. I think it was just a function of the different conditions used to take that picture. I assume those were taken in a lab. So I don't know, is the short answer. And then the last one that was, the last question that was specific to the lab activity itself was between feeder and non-feeder systems for growing stem cells. So feeder systems were kind of the first ones people used, and it's very robust. It works often, which is important when we're doing stem cell research because a lot of times it's not very efficient. But feeder systems actually use cells from mice to provide that support for stem cells and some of those growth factors and other nutrients. So it's helpful when you're doing basic research in the lab when all you need is just some cells to be alive. However, when you're studying things that will be used for treatments or for clinical applications, having a feeder system can introduce some unwanted things. So you don't want any contaminants like viruses or bacteria or other things coming from those mouse cells. You don't want to inject those cells into a human being. And so that's why people develop feeder-free systems, which don't have other cells. It's just the stem cells you want to grow with the specific combination of support and nutrients. So vitronectin was the one you used in the lab. There's a couple different other types of nutrients and supports that people use to grow cells. And it's used for clinical research because then you're not introducing any other biological factors. So that someone had a question about why wouldn't we use like the equivalent of fibroblasts. And again, you want just the cells from the patient specifically, so no cells is a cleaner option. So those were the ones specific to the lab activity. I'll answer a few of the other ones kind of as we go here. So stem cells in general are cells that not only can replicate themselves, but are able to differentiate into other ones. So there's a few different flavors. The first cells are totipotent, so they're able to differentiate into any kind of cell. So embryonic cells as well as those 3 tissue layers. So this is a figure in your book kind of augmented. Even though they start out looking extremely similar, each of these cells or embryos will develop into very different organisms. And that's determined by transcription factors and epigenetic factors and all kinds of other things that lead to cell differentiation. So totipotent are the ones that can become anything. We'll see in this diagram the different flavors. So embryonic stem cells, right, start as totipotent and then develop into this blastocyst stage of the embryo. So when people research embryonic stem cells, they're taking this population here from the embryo. There's also induced pluripotent stem cells, which you guys saw in the lab. So instead of taking them from embryos, you're actually able to take skin cells and then give them certain proteins that tell them to kind of switch and become reprogrammed, in other words, so that they can differentiate into other things. So induced because we're actually changing that cell behavior. We're going back from a differentiated cell into one that can become many different kinds. So someone asked the difference between pluripotent and multipotent. So pluripotent stem cells can become any of those 3 different cell layers, whereas multipotent have differentiated to a certain degree and so only can become certain other ones. So blood cells are the ones that have been studied the longest in terms of stem cells. So a hemopoietic or a blood stem cell is multipotent because it has differentiated to the point where it knows it's going to be a blood cell of some kind, but it can still become many different kinds of blood cells. So multipotent has kind of narrower options than a pluripotent stem cell. And then a unipotent stem cell has one job. It's going to change into one thing. So we've ranged from totipotent, which can become any kind of cell. We've narrowed it down to pluripotent stem cells, which have still lots of options. Multipotent stem cells have a couple options, and unipotent have one. And so here's the link to the article where this diagram came from if you want more information. Okay, so stem cells, very helpful. We need them to differentiate into our different tissues, right, to become that sea urchin or a mouse or a human being. So very important to know their biology, but also because we can use them to treat disease. And so because I love plants, I needed to show you an example of plant stem cells. So if you think about how roots are growing, there's one little tiny bundle of stem cells in the plant root called the quiescent center. So in here in this diagram, they're gray. There are just two tucked in behind the root cap. And from those two stem cells grow all of the other cells that become the roots. So plants have stem cells in different places, just like humans. So in our case, we have in our gut epithelial cells, tissues, these painted in red right here are stem cells that continually replenish that lining of our gut. So a lot of tissues, a lot of epithelial tissues are continually replenished. And to do that, we need stem cells. So kind of in the little crypts right here is where we find those for our gut epithelium. And then in this diagram, you can see, again, from that quiescent center in plants come all of these different tissue types. Okay, so plants have stem cells, animals have stem cells, and if you want to see a really cool example of a model of a root growing, you can check out this link right here. Okay, so they're in different places in your body, used in different points in development. So what can we use them for? Well, your book kind of mentions two applications of cloning. So we didn't get to the cloning lab for this class, but these are two examples of what that looks like. So when we're thinking about using stem cells to treat diseases, someone asked about skin grafts. I don't know if the technology is there yet, but it's a really hopeful possibility, right? If you have really terrible burns that cause a lot of damage, that damage is permanent. But if we had a way of treating those injuries with stem cells, we could regrow healthy skin, and we wouldn't have that lasting damage. So I don't think we're there yet, but people, I'm sure, are working on it. But one of the ways we could possibly do that is taking cells from adult tissue. So I don't quite remember what specific tissues those could be, but I know skin fibroblasts are one candidate. So you take that cell, it has all of that genetic information you need, like all of your cells do. You can take an unfertilized egg from another person, remove the nucleus with a really, really tiny capillary needle pipette, and then you can inject or fuse an adult cell into that egg cell that no longer has a nucleus. So it contains all of that information and programming that would have made it a totipotent cell. You've introduced a patient's genetic information, and so you can then have that embryo be of the patient's genetic information, even though it's an egg cell from someone else. And so that's the technology behind reproductive cloning. So Dolly the sheep or other animals are cloned this way. but this is also a way of making personalized embryonic stem cells. So there's still some ethical issues around harvesting treatment of these cells, but it's one option that scientists can pursue. And so because it has your own genetic information, there's a lot less of a chance of rejection or complications with incompatible genomes. So 2 different ways of cloning. This was also in your book kind of as an example of how the general public or people not familiar with how cloning works can misperceive what stem cells are useful for. So anyway, just an overview of how we can make personalized stem cells. So these are some of the technologies behind making model systems like we saw in our lab. So we'll go on to one more example of making stem cells. So induced pluripotent stem cells were cells that had already differentiated, but we can trick them into becoming more like pluripotent stem cells, which can still arise to multiple different tissue types. So these, again, are from your skin. So you take a skin sample, grow them in a petri dish, and then there are 3 specific transcription factors that are needed to induce this reprogramming. I meant to put the scientist. It was another scientist in Japan that discovered this. So you can Google that if you want more information. But then this induces this pluripotent state, which can then become neurons, heart muscle cells, etc. Okay, so this last slide just covers the rest of the questions I had from you guys. So someone asked which diseases are pluripotent stem cells used to treat most often. And although there's tons of applications for what we could use them for, like spinal cord injuries and any other kind of tissue damage that is permanent now, the most common treatment are for blood cancers. So a bone marrow transplant is actually the most common stem cell therapy. Blood cells are the most commonly studied. They've been studied the longest. That's where most of the technology is now. So if you want more information, you can check out this link. And someone asked who first identified stem cells. And the people who were the ones studying blood cells and how they arise were Ernest McCulloch and James Till and their lab members that contributed to that research. So there's a summary link right here, and you can find from that link the papers that describe those first definitions. And then someone asked, so the whole premise of the lab was using stem cells as a model for Parkinson's. So how are these stem cells used for that? Well, you can grow neurons, human neurons, in a dish, and you can use those to study how they behave and how they react to different drugs. So if you're thinking about finding a treatment for Parkinson's and you have something and you're not sure how it's going to interact in the body, you can start by growing cells on a dish, adding the drug, and just seeing how they react. So of course there are later clinical trials, but it's a really good way to screen lots of drugs at a time in a way that doesn't harm human health. Okay, so that was stem cells in a nutshell. There's so many new findings and new therapies. It's a hot topic of research, so I encourage you to learn more about that if you're interested. But I think that covered everyone's questions from the lab. So with that, I'll transition to cancer. So I'm really interested in the history of words, so I looked up why a disease is named after a crab, and it's because those scientists and medical professionals of antiquity thought that name fit. So Hippocrates, pretty famous philosopher, scientist, person in Greece, named cancers carcinos, or crabs. There's a couple different explanations people have come up with for why. It could be the hardness, the firmness of tumors when you feel them through your skin. It could be the pain is reminiscent of a crab pinching you, or the pervasiveness of cancer in that it doesn't let you go like a crab holds on tight. But whatever the reason, he named it after a crab, and the Greek word is karkinos. A few hundred years later, a Greco-Roman philosopher named Celsus found the writings of Hippocrates, and this person was compiling a medical encyclopedia, and so he translated it from the Greek to Roman, and Roman for crab is cancer. Then another 100 years or so later, Galen, another famous historical medical person, translated or noticed that when he was dissecting someone's breast tumor, it was spreading in a way that reminded him of a crab's leg kind of extending outward from the central tumor. And so that's why it's stuck. So these are pretty renowned people, so their writings have persisted through the ages, and so that's why cancer is called cancer. And coincidentally, someone who studies cancer is not called a cancerologist. They're called oncologists because onkos is Greek for mass. And since a lot of cancers have these tumorous masses, oncologist is just the study of masses. So now that you have kind of the historical context, we can go into what it means. So of all the things that kill human beings, at least in the U.S., cancer is the second most common cause of death. And so you can break these down into the different categories. It follows right behind heart disease as the most common leading cause of death. And it's far out ahead. So the two most common causes represent a lot of causes of death. And if we break down the different kinds of cancers, we can look at the ones that are diagnosed the fastest, or the rate is higher. So breast cancer in females has the highest incidence of new diagnoses, all on down. And cancers can range in severity, so things can be really swift and devastating. There are some cancers, I think it's a thyroid cancer, that spreads so slowly that a lot of people die without having ever realized they had it. And in fact, treating some of those is sometimes worse than the disease itself. But more often than not, they're going to grow over time, and if not very quickly, then be devastating once they get large and spread. So the ones that are diagnosed at higher rates are not necessarily the ones that are the most deadly. And so the factors that differentiate those are a few, but especially ones that are hard to detect. If it's allowed to grow longer, they're harder to treat and are often more deadly. So what actually causes our healthy cells to turn into cancer? We'll get to that after we get to some terms. So cancer can range in severity. Sometimes cells will grow and divide but won't spread. So a tumor is benign if it's showing no evidence that it's invasive. So those you can surgically remove and have a really good prognosis. If a tumor is malignant, then it's invasive. It's getting into other tissues. And that's important because if it moves, if it spreads, it can form a metastases or metastatic tumors. And that's when they go to different places in your body and proliferate there. So those are the really deadly ones, the malignant tumors that have metastasized, that have moved around. And then when we're thinking about different kinds of cancers, they're named after the tissues of origin. So carcinomas occur in skin cells, epithelial cells. Sarcomas come from muscle. Leukemias are certain cancers in your blood cells, nervous system tissue. Cancers can arise in all of these different ones. But the ones we see most often are in cells that replenish themselves often, so they're already kind of programmed to grow. And so a lot of epithelial cells give rise to certain cancers. So what turns a healthy cell into a cancer cell? It takes a lot, actually. But so different forms of genetic instability will lead to cancer, and that looks like lots of different things. But at the end of the day, once we interfere with 3 specific pathways, you're going to lead to cancer cells. So this is an image from your book. And once we get mutations or changes in genes that are in these 3 different pathways, in cell proliferation, DNA damage response, so that p53 protein was produced in response to DNA damage, and if we had p53 active, it led to this cascade that led to apoptosis. And so if we have DNA damage, we don't want to reproduce those cells. But if we have cells that are already growing when they shouldn't, they can accumulate more DNA damage. They can grow more, use more nutrients. Then we have cancer cells. So there could be hundreds of different mutations within these 3 pathways, but once these 3 break, you can lead to cancer. So thinking about cell proliferation and apoptosis, if you're able to kill cells that need to die, great, you can have healthy tissues. If you have cells that start to grow too rapidly, you can have apoptosis still function, but because those cells are growing too much, they're dividing too often, you can get a tumor. So this might be a scenario where we have some benign tumors. You can also have a situation where you have normal cell division, but if you're not able to kill those cells that have something wrong with them, you can also lead to a tumor. So in a specific instance, again, blood cells are the most commonly studied. So our initial blood stem cells are here. They're able to regenerate themselves. They can produce another type of cell. So kind of different degrees of differentiation. If this cell that's on its way to becoming a red blood cell is stopped from furthering its progression to the right kind of cell, it can form a leukemia because it's growing uncontrollably, it's not developing properly, so they can accumulate more and more mutations. So a lot of different mutations, again, can lead to cancer. Once we get rid of the ability to check for DNA damage, then those cells can then accumulate even more changes. So we can lead to some pretty gnarly karyotypes. So this is a table from your book as well. So there's a couple factors that can lead to this instability in cancer cells. So if we can't replicate properly, if there's defects in the pathways that repair DNA, those, if the cell is healthy otherwise, will be checked at those different points in the cell cycle. But if those are broken, we can't check for DNA damage. It will be replicated without fixing it. That leads to mistakes in mitosis. And along the way, then we can have abnormal chromosome numbers. So we might carry more than one sister chromatid into one. There's lots of factors that can occur in meiosis, a lot of errors that can happen. So just a summary of the different types of things that can lead to the instability that can lead to cancer. So I showed this a few weeks ago in class, but this is a picture of the karyotype of some of the most famous cells on Earth, so HeLa cells. So these are cells that arose from a tumor that came from Henrietta Lacks. And so these cells are able to live on in perpetuity. So she died from her disease in the 50s, I believe. But scientists, doctors, took her cells without her permission and studied them. And this cancer is so pervasive that people that are studying different cell lines have had their cultures kind of contaminated by these cells, and they just take over. Cancer cells become very aggressive, and I'll get into that in a few. But from the karyotype, you can see not only are there not the appropriate number of different chromosomes, but they've switched. They've taken some information from others. So we have this bit of chromosome 7 is stuck to an extra chromosome 16. So lots of different aberrations. This is a very unhealthy karyotype. But all of these changes have accumulated and led to its ability to survive for so long. So if you're looking for summer reads, I highly encourage you to read The Immortal Life of Henrietta Lacks. It goes into her background and history and issues with ethics in medicine and things like that. So things we've been talking about in class. So these different mechanisms can break down. We can accumulate these mutations. It leads to really big rearrangements in chromosomes, which has devastating effects for our health. So I'm going to go over a couple different ways we can get mutations that cause cancer. So there are dominant mutations or gain-of-function mutations. Right? So if we remember to genetics, dominant mutations, we just need one chromosome to be affected to see a phenotype. So these are called oncogenes. Again, oncos means mass. So these are genes that if they have these dominant mutations can lead to cancer. When they're in a normal cell and they're undamaged, they haven't mutated, they're called proto-oncogenes because we see a lot of the same ones getting certain mutations. So proto-oncogenes are perfectly fine, code for lots of different proteins, but once they get that mutation, that gain-of-function dominant mutation, they become an oncogene. So mutations in these genes lead to proliferation of cells when they survive when they should be undergoing apoptosis for lots of different reasons. Dominant mutations, oncogenes. We can also get cancer from recessive genes. So in this case, these genes are called tumor suppressor genes. So when they're healthy, when there's no changes, they're able to suppress the growth of tumors. However, after we've accumulated two mutations, so in both of these chromosomes, in both of these genes, they're no longer suppressing tumor growth. Tumors are able to form and proliferate. So dominant mutations are oncogenes. Recessive mutations, we need two to negate those tumor suppressor genes. And so there's lots of different ways that we can that we see these mutations occurring. So we might just have a base pair change, a codon change that can lead to a protein that's overactive. And these are those gain-of-function mutations. So we're going from a proto-oncogene to overactive protein. So within the sequence, we can make a change that leads to an overactive protein. There's also mutations where we have too many copies. So if we have some kind of breakage in the ability to repair DNA, we can have multiple copies, and that leads to an overexpression of the amount of protein, so there's too much around. But there might also be a rearrangement. So we saw in that HeLa karyotype that there were pieces of different chromosomes mashed together. So if we have a situation where there's a regulatory sequence right by that will always turn this gene on, we can make too many protein products. Or if there's some other protein or regulatory element that can fuse to that protein that makes it hyperactive. So it's something that changes that protein structure itself that can increase activity. So these are all examples of how we can turn a proto-oncogene into an oncogene, that gain of function mutation. And when we're looking at tumor suppressor genes, those recessive mutations, loss of heterozygosity. So if we have too many homozygous regions in our DNA, it can lead to needing only one recessive allele. And so if we are thinking about inheriting one gene where we've lost that function, so we have one tumor suppressor gene that's active, that's actively working against cancer forming, but we have one that has a mutation. We're closer to getting the ability to form cancer. So over the course of someone's life, something might happen where that normally functioning gene gets silenced. So even though the gene itself is active, some other kind of epigenetic change can silence it, and now you have two defective copies of a tumor suppressor gene. It's no longer going to protect against cancer formation. You can also gain another mutation in that other one, or have that section deleted. Lots of different ways where you can get these different mutations that lead to cancer. So kind of thinking about these different types, looking at how it can progress. Let's say we start with our healthy normal cells. We lose a tumor suppressor gene, so we have two of those recessive mutations. Now we have a case where we have these cells accumulating. There are too many growing. We can handle that. It's not a problem yet. But then one of these cells in this population has an oncogene, so some gain of function mutation. So let's say a ROS pathway is activated. Well, that can lead then to a small tumor. We might not even be able to feel it's there. But if another tumor suppressor gene is lost, we're not able to keep that cell in check anymore. That tumor can become larger. Another tumor suppressor gene can be lost, in this case p53, which again helps with DNA damage and apoptosis. So that can convert that large tumor from being benign to being malignant, so it's able to spread. And once it becomes invasive, it's accumulated so many mutations that prevent it from fixing DNA or checking its growth. It can ratchet up the number of mutations it accumulates, which can lead to it spreading in your body. So even if we lose one gene, even if we're that unlucky recipient of two loss-of-function genes, on its own, it's not going to cause cancer. We need to accumulate many of these certain mutations in order to lead to disease. So we'll see what that looks like in kind of picture form. So we have this initial cell that's not doing what it's supposed to. It's not checked in its growth. It can grow, become a benign tumor. It accumulates some kind of mutation that either an oncogene or a tumor suppressor, and it's able to invade. And once it can get into blood vessels or your bloodstream, and it can travel and lodge into many different tissues. And everywhere it spreads, it has more opportunities to accumulate mutations. So when scientists have looked, and doctors have looked at, medical doctors have looked at how many, so kind of like how many licks does it take to get to the center of a Tootsie Pop, how many mutations does it take to form a metastatic cancer, the average is between 7 to 9 of these dramatic genetic changes. And having one or a couple of those increases the likelihood that you'll gain more. So why does that happen? Why are certain cells able to accumulate these mutations? Well, cancer cells, the populations of cells actually evolve. So once you have cells that have these different mutations, right? Natural selection acts on the DNA, the traits. If these cells are able to grow and outcompete their neighboring cells, they're more likely to survive. So they're actually selected for within an individual organism. They have different metabolisms. They're able to produce new blood vessels. They're able to change very quickly and can look very different from your normal healthy tissues. And one of the reasons cancer is so hard to treat, even though its occurrence is highly unlikely when you look at the face of it, is that once it starts, it's just going to keep growing. And often cancer isn't detected until it can be too late. So thinking about exponential growth, it's undergone so many doublings of its population that by the time it's first visible on an X-ray, you already have thousands and thousands of cells. So if it's spreading and growing, it can be hard to surgically remove, and that's where we look at different treatments. So we have all of these new technologies. There's lots of ways to treat cancer. So the fact that sometimes we don't detect it in time is one reason it's hard. But another reason it's difficult is that there's just such a vast array of possibilities of getting cancer that a treatment that works for one may not work with another. So we'll look at different ways of treating cancer in these last few slides. So as a summary of what we do, the different types of approaches, starting with surgery, just physically getting it out, getting rid of those cells so they can't proliferate anymore. Sometimes this is a treatment in its own, and you can remove the entire tumor, especially if you detect it very early. So if we look back at this diagram, the chance of getting it all, especially if it's very invasive, is lower and lower as more doublings of these cells have occurred. So surgery is often done in combination with other things. So radiation therapy is using radiation to change the DNA in tumor cells so much that they just can't survive. So this is in combination with either surgery, or it can be in combination with a couple different other treatments. Chemotherapy, treating cells with different chemicals to try to kill those cancer cells. These are all really intense procedures that have a high chance of damaging other cells, so there's lots of side effects associated with these, and it's all different, again, depending on the person and the area where the tumor is. There's other therapies that are coming out, immunotherapy, like using your immune system to attack cancer cells, which has the benefit then of not using such harsh treatments that will harm your healthy populations, using your own immune system to go after what it knows is not healthy tissue. There's targeted therapy, so more and more personalized medicine now that we have so many good genomic and genetic tools. And there's also stem cell transplants. So like I mentioned, the treatment for a lot of leukemias and other blood cancers is a bone marrow transplant. We're looking into more ways of using stem cells to treat these cancers. So I'll go into a couple different types of these immunotherapy ways of treating cancer. So there's something called immune checkpoint therapy. So as the cell undergoes its different stages, right, there's different places where the cell is checked to make sure it can proceed. So I haven't read this paper completely. If you're interested in this technique, you can go to the link right here and read about the different proteins that contribute to this. But for my purposes, just knowing that checkpoint therapy is something that you can use. And one of the hottest new techniques that people are looking into is called chimeric antigen receptor T-cell therapy. So chimeric is a word that means a kind of like a mashing together of different components. Antigen receptors are just things that recognize other proteins, and we're putting this chimeric mishmash of things on our own T cells, which is part of our immune system. And so this is one of those promising immunotherapy treatments. And what that means is that in a patient that has cancer, scientists will remove some blood to gain some T cells. In the lab, they will be able to insert the gene that codes for this specific receptor. And so then those cells are able to grow with this cell protein that recognizes a certain other protein. You're able to grow millions of these cells in the lab, infuse them into the patient, and then those T cells, that part of their immunity, has those receptors that will recognize a protein that's expressed only by the cancer cells, and will be able to recognize those as invaders and engulf them and take care of them. So people are looking at this with lots of different target proteins in mind. You have to be really careful when you're selecting them. You don't want to have an antigen receptor that will bind a protein found on healthy tissue, because then you're using your immune system to attack itself, which can result in some really bad complications. So researchers are working to find specific proteins that are only expressed and are expressed a lot by cancer cells, so that these cancer cells can't evade this detection. So in this diagram, you can see a couple different examples of what people are using to try to target. And if you want more of the background information, you can find the publication at the link. So these are showing a lot of promise. We're advancing all the time with different areas. And again, there's so many mutations that lead to cancer. We're looking at it in all kinds of different directions. So this is a really good area of medicine to work in that addresses a lot of human suffering. Again, the second leading cause of disease in the U.S., the chances of accumulating these mutations are increasing as you get older. So in countries that are really wealthy, you see high incidences of cancer, especially later in life. So that's all the information that I have for you guys. Thank you so much for giving me such an amazing semester at Illinois College. You guys have been on top of things and have worked so hard and asked such good questions. You keep me on my toes. So from the bottom of my heart, thank you for being such amazing students this semester. I hope you're well. I hope you finish the semester strong, and if you're graduating, congratulations. I'm looking forward to seeing what good you guys do in the world. Thank you so much.